Okay, grab your coffee, get comfortable, and let's map out the absolute chaos that is a cell under stress. Welcome to Patho 101, the ultimate Unit 1 survival explainer. We're taking all those incredibly dense concepts from your first unit and, transforming them into a visual roadmap so you can completely crush your upcoming exam. Let's dive right in. So here's our game plan for today. We're hitting the framework of pathophysiology, cell adaptation, injury, and death. Then we'll look at genetics and congenital errors, what happens when cells go completely rogue, and wrap it all up with immunity and inflammation.
Section 1. The Framework of Pathophysiology, Building the Foundation.
Okay, the absolute crucial starting point is remembering this exact sequence. It's the big four. Whenever you look at a new disease, ask yourself, first, etiology. Basically, why is this happening? Is it a genetic glitch or maybe something environmental? Next is pathogenesis. How exactly does that initial trigger mess with the cells over time? Third, clinical manifestations. What do you actually see when the patient walks into the room? And finally, all those earlier steps literally dictate the fourth pillar, which is your treatment implications. Fix the why and the how, and you know exactly what to do for treatment.
Now, let's zoom in on that third pillar for a second. Clinical manifestations. It is so easy to mix up signs and symptoms, but here's the trick. Signs are objective. They're the hard evidence you, as the clinician, can actually measure. Think of a fever on a thermometer or a visible rash. Symptoms? Those are entirely subjective. That's the stuff the patient has to tell you about, like feeling nauseous or having a killer headache. You can't measure a headache, but, it's definitely there.
Section 2. Cell Adaptation, Injury, Death. The Journey of a Cell Under Stress.
Check this out. This is our very first red flag, reversible cell injury. When a cell gets stressed, say it's choking from a lack of oxygen, it stops making ATP. Without that energy, the sodium-potassium pumps just quit. Sodium builds up inside, and water immediately rushes in to follow it. It's kind of like a sinking ship taking on water. The result is hydropic swelling. You can actually see it in those pale, super swollen, blebbing kidney cells on the right, especially when you compare them to the healthy ones on the left. But hey, good news. If you restore the oxygen in time, this whole process is entirely reversible.
All right, let's do a quick knowledge check to test your active recall. Imagine you have a 55-year-old patient who has been smoking for 30 years. That chronic stress from the smoke actually forces their delicate, ciliated airway cells to swap out for tougher, flatter epithelial cells. What do we call that specific type of cellular adaptation?
Boom! The answer is metaplasia. It's the reversible conversion of one adult cell type to another. The body is honestly brilliant. It realizes those fragile original cells just can't survive the constant toxic smoke, so it swaps them out for a cell type that can actually handle the persistent injury.
But what if the injury is just too severe, or it goes on for too long? Well, it crosses the line into irreversible territory and the cell dies. This goes down in one of two ways. First, necrosis. Think of necrosis as a messy, chaotic explosion. It's triggered by external injury, the cell violently ruptures, spills all its guts, and kicks off massive inflammation everywhere. Then you have apoptosis. This is neat, highly programmed cell suicide. The cell turns on internal signaling cascades and quietly dismantles itself without rupturing. Zero inflammation. So chaotic explosion versus a highly controlled demolition.
Section 3. Genetics and Congenital Errors. The blueprint goes wrong.
Now look at this. Right here, we're seeing the exact microscopic mechanism of a point mutation. See how swapping just one tiny letter, literally a single nucleotide in the RNA sequence, completely changes the amino acid that gets built? It goes from a normal lysine to a totally mutated state. Just one tiny spelling error in the genetic code can scramble the entire structure and function of a protein. It's wild. It's a perfect illustration of how the smallest molecular changes can cause massive, systemic inherited diseases.
Section 4. When cells go rogue. Hematologic neoplasms.
Honestly, the most life-saving thing you can take away from this explainer is knowing how to spot a blast cell under a microscope. These guys are the main culprits in acute leukemias. Picture this, a highly immature, malignant white blood cell with a totally massive nucleus that hogs almost the entire cell. You can see those visible nuclei, the lighter spots inside, and there's absolutely zero cytoplasmic differentiation. It's just a raw, primitive cell multiplying out of control instead of growing up and actually doing its job.
Now, look at the striking visual difference between these two malignant disorders. On the left, we've got Hodgkin lymphoma.
Do you see that massive cell right in the middle, staring back at you with those two dark purple circles? That is the classic binucleate Reed-Sternberg cell. People always say it looks like owl eyes. Spotting that cell surrounded by other normal immune cells, that's the definitive hallmark of Hodgkin lymphoma. Now contrast that with the right side. That is a dense, overwhelmingly crowded sea of malignant cells, just infiltrating the tissue and completely wiping out the normal architecture.
Pop quiz time. Let's do another quick check-in to make sure we've got our foundational terms locked in. Which of the cellular adaptation trophy brothers describes a situation where cells increase in actual size because of a higher workload? Think of your heart muscle having to pump against high blood pressure for years. Yep, you guessed it, hypertrophy. Hypertrophy means the existing cells literally get bigger and bulkier. It's exactly like a bodybuilder's muscles bulking up in response to heavy lifting. And that is completely different from its brother atrophy, which you can see on the left. That's where cells actually shrink and lose substance because of disuse or poor nutrition or lack of blood supply. Remember, in hypertrophy, you aren't making new cells. The ones you already have just get supersized.
Section 5. Immunity and inflammation, the body's defense systems.
This right here beautifully captures our innate immune response in action. When tissue gets injured, like that cut through the skin at the top, the damaged cells send out molecular SOS signals. Check out the blood vessel at the bottom. It undergoes vasodilation and gets way more permeable. This literally lets our heroic phagocytes squeeze right out of the bloodstream and rush straight toward the invading microbes to clean up the mess.
And because you have this massive rush of fluid and inflammatory cells flooding the area, you get the 4 classic physical signs of acute inflammation. That vasodilation brings in extra blood, which causes redness and heat. The leaky vessels let fluid seep into the tissues, causing swelling. And then, that swelling, combined with all those chemical mediators, starts pinching the local nerve endings, which gives you pain. So every single symptom has a direct physiological cause. It's basically a chain reaction. But sometimes our amazing defense system kind of overreacts, and that leads to hypersensitivity. There are 4 types, but let's focus on type 1. Type 1 is your immediate, classic allergic response. We're talking minutes here. It's driven by IgE antibodies locking onto mast cells. The second you're exposed to an allergen, those sensitized mast cells instantly degranulate. They just pop, dumping massive amounts of histamine everywhere, which leads to things like allergic asthma, rhinitis, or even life-threatening anaphylaxis.
Wow. Okay, we have covered a massive amount of ground today. We went from the core framework of disease all the way to metaplasia, rogue blast cells, and the inflammatory cascade. So, I want to leave you with this final thought to really ponder before you tackle Unit 2. If we know exactly how these cells adapt, how they mutate, and how they inflame at a super precise microscopic level, how do you think we can hijack those exact same molecular mechanisms to design drugs and cure them? Let that sink in. Study hard, and you are absolutely going to ace this unit.